More recently, we've been exploring this for monitoring responses to vaccination. So since this is in thermodynamic equilibrium, we can have the sensor, we add the RBD and it opens. Now, if we add serum samples that contain antibodies against the RBD, they compete with the sensor and we get a loss in signal. Um, and we actually have nicer data than this now, but so we've been looking at samples from patients who have been vaccinated who have different levels of response to the virus, and we can assess their antibody response by looking at the neutralizing antibody titer, and we can also look at the change in signal. You can see they're quite well correlated. And so this is a very quick test you could run at home, read on a cell phone, and so we're hoping this will be useful. We can take the same concept, and I showed you we can design binders to many other types of proteins. And so we can convert these into sensors in the same way by caging them in the off state of the device, so that the protein device, so that when the target is present, the thermodynamics drive opening. And so, for example, this is a troponin sensor. In these experiments, what we've got many different sensors, and then we're adding to each one the target. And this is similar to the specificity plot I showed you before. Only when we add a troponin to the troponin sensor do we get the light emitted. And here's, for example, one against the botulinum toxin, where only the botulinum toxin sensor is responding. So we think this is sort of a very general way of making molecular sensors. And again, I wanted to give you an example of how we can use this sort of molecular device concept for problems sort of beyond coronavirus, and in this case, beyond sensing. And here, this is a rather different problem, at least on the face of it, the problem of doing calculations in the body, because antibodies just work by going after specific targets. But sometimes you want to do more subtle things, like you want to recognize cells that have two markers, but not just one alone. So you may have a tumor that doesn't differ from healthy cells in any single marker, but differs in the combination. So we can take the same type of switchable system and direct the different components to different cell surface proteins. And so only when we reconstitute the full system do we get switching. And in this case, rather than emitting light, what we're doing is we're exposing a motif that recruits an effector, for example, a CAR T cell. And that example is shown here. So here, this is a little bit more complicated. So imagine a tumor cell, which has two markers which only occur together on the surface of the tumor, and then it also has a healthy cell marker. And we want to make sure that we don't activate CAR T cell signaling of any cells that have that healthy cell marker. So then what we do is we take the two components of the switchable system that I mentioned, that I described earlier, and direct it to the two components which occur on the tumor cell. And then we take a third design, which actually sequesters one of those two switching components required for switching and direct it at the healthy cell marker. And if we do that, then we only get CAR T cell recruitment and activation when in the presence of cells that have the two tumor cell markers, but not the healthy cell marker. So that's here. And the third area that I wanted to tell you about in context of COVID is vaccines. So for a number of years now, we've been developing methods for designing self-assembling proteins, and much of the rest of my talk is going to be about self-assembly. And in particular, we have been developing a wide variety of polyhedral nanoparticles. And so the basic principle is to match the symmetry axes of a polyhedron. For example, in this case, an icosahedron, which has 5-fold and 3-fold symmetry axes, we place protein homoligomers with 5- and 3-fold symmetry on those symmetry axes, and we can move them in and out and rotate them, and then we design interfaces between them. And using this procedure, we've been able to design a wide variety of very homogeneous protein nanoparticles. You just express the two components in E. coli, they self-assemble, and these are just EM fields of what comes out. And so they're very homogeneous and robust. So my colleague Neil King at the Institute for Protein Design has taken the receptor binding domain of the spike and then displayed it on the surface of these one of these nanoparticle systems. And again, he gets very homogeneous nanoparticles. You can see the little RBDs, relatively little on the surface here. Now, what's very exciting is that these induce very, very strong responses. They're much stronger than to the trimer alone. And this is because avidity. We think it's because avidity is really stimulating B cell that have the corresponding B cell receptors. So the current mRNA and other subunit vaccines basically use the trimer, and now with these nanoparticle vaccines, there's considerably more neutralizing antibody elicited and better protection. Oops. So these actually have been in clinical, Neil's team has these in clinical trials for the last 6 weeks, and we're about to get feedback on them. These are among the, the most promising of what are known as the second generation coronavirus vaccines. So now we've, again, going beyond COVID, we've been sort of extending this concept of building nanoparticles from proteins with symmetry. And what we realized is that we could actually combine form and function by embedding symmetric proteins in these nanoparticles that actually have functions in addition to symmetry. And here we're backsliding a little bit because one of the proteins we're using is actually a naturally occurring protein. I think that's the first time in my talk where that's happened. And so these yellow things are antibodies. And so what we've done is to design, in this case, a pentameric protein which binds the Fc region of an antibody such that the 5-fold axis of the pentamer and the 2-fold axis of the antibody lie on the 5-fold and 2-fold symmetry axes of an icosahedron. So what that means is if you take this designed pentamer and add it to any antibody, it will assemble it into a perfect icosahedral nanoparticle. We've also made C4 proteins tetrameric proteins that hold antibodies so that they're placed on the 2-fold axes of icosahedra. And here we have a trimeric one that holds antibodies, so they're on the 2-fold axes of tetrahedra. And so you can take any antibody and rapidly format it in these different oligomeric states. And then you can look at the effects of the antibody, for example, on signaling. And this is an example with an anti-death receptor antibody, where we can basically this antibody alone doesn't do anything to tumor cells, but by changing the presentation of the antibody changes the engagement of the death receptor, and we get killing of the cells. So if you have any antibodies you're interested in and you'd like to potentiate or explore how clustering of their receptors alters what the effect of that is on signaling, now you can do it very easily. There's no covalent modification or anything else required here. It's just non-covalent interaction. So beyond biology, I just wanted to show you sort of kinds of things that we're thinking about. So, and this is happening a lot in the group now. Nate Ennis designed a C2 protein that binds 2 chlorophyll positions, as in the special pair in photosynthesis. And then those have very interesting optical properties on their own. And then Shunji Wang built these C2 structures into octahedra. And so now we have octahedra with a C2 chlorophyll pair on each axis. And this is actually just showing you, again, this is a case where the design is very close to the computational model. So we're now thinking about putting electron acceptors in the middle, like nanoparticles, to try and start creating artificial light harvesting complexes. Okay, so now this is where I start talking about all the fantastic things we've been doing with Manu and his student Joe. So this is work by Ariel Ben Sasson, and so Ariel took a D2 protein and a D3 protein and arranged them so that they would come together to form a hexagonal lattice. And this is what happens when you produce the proteins in E. coli. You get this very nice lattice, and he put a GFP in at the trimer vertex, and you can see the GFPs there. And so then Ariel made the proteins separately, and when he mixed them together, they spontaneously form this really beautiful and regular hexagonal lattice. And this is a superposition with Ariel's design model. There's the D3 and the D2 components. They're exactly where they're supposed to be. So Ariel, in a great good fortune for my group, met Amanu at a meeting a few years ago. And we've had this really exciting collaboration where Joe and Manu have been functionalizing these components and getting really spectacular results, which I'm hoping you've all heard about, so I'm not going to go over them again, in being able to demonstrate assembly on cells and very interestingly, these arrays shut down endocytosis, and they have really profound effects on cell polarity, which if you haven't heard Manu speak about, you definitely should. It's absolutely amazing. So we've also been thinking about in nature we have proteins that interact with inorganic minerals and cause mineralization, as in tooth and bone and seashells. What if we could design proteins that could template inorganic material mineralization? But what if we could do it with semiconductors and other materials that are of interest to modern humans? It would be really cool. So we've been starting to look at this. So this is work from Harley Piles. He designed proteins which are perfectly epitaxially matched to an inorganic surface. So they're repeating proteins that have a repeat spacing that matches, in this case, the mica lattice. And he made these proteins long enough so they were visible by atomic force microscopy. You can see them binding here. And then what he did was design interfaces between these to direct assembly of different types of assemblies. And this is now a hexagonal lattice. Again, it's different from one that Ariel designed because this is only forming on mica. So these individual elements, proteins, are epitaxially matched, and then they have interfaces which direct formation of this basically trimer on the mica. But since these are perfectly straight proteins, Harley could make a series of variants just by making them shorter and shorter and shorter. This doesn't really work with naturally occurring proteins, but you can see the experimental results down here. The hexagonal lattice gets smaller, finer and finer, and these are averages down here. So what we're now trying to do is to go flip this around and use the designed proteins to actually template inorganic mineral growth. Okay, so everything I've told you about so far is soluble proteins, but we can take the same principles and apply them to membrane proteins. And this is work of Chunfu Zhu. He designed these alpha helical channel-like proteins, which have 2 rings of alpha helices surrounding a central pore. These are soluble protein designs, and these are comparing the design models to the crystal structures. So he and Peilong Liu then took these and they resurfaced the outside to make them membrane compatible and expressed them in mammalian cells. And what was very exciting, this in collaboration with Bill Catterall, is that these proteins turned out to be very selective potassium channels. So this is looking at conductance. This is now a patch clamp on a mammalian cell expressing this protein. This is looking at conductance for potassium. You can see it's much more selective for potassium than sodium. And this is kind of interesting. There's been so much written about what makes potassium channels selective for potassium. And here we could just design a protein with a pore and get potassium selectivity pretty much right off the bat. Now, what we can do now is, and we're doing is we can start exploring the effect of the exact composition of the lining on the ion conductance. In this experiment, Chunfu took a glutamate that was near the entrance of the channel and turned it to a cysteine. And what he found then is that, it's easiest saying here, is that this channel still conducted ions just fine, but if he chemically modified that cysteine, the conductance went away, sort of demonstrating that the ions are actually flowing through this central channel. Now, Peilong and Chunfu also, I told you there were those larger channels, the ones with the 16 helix, and those, this is a cryo-EM structure Peilong solved, and there's a really big channel here, which actually allows small organic dyes to go through.